This is Organismal Biology, and I am your instructor, Dr. Jibo Zanzo. When I first learned that I was going to teach this class, I wasn't sure what I was getting into, so maybe you feel the same way. Here is the deal. This course follows on Biology 1020, Principles of Biology, which introduced many concepts that are found in all or nearly all living things considered to be alive. Things like the essential types of molecules, carbon, water, proteins, lipids, carbohydrates, nucleic acids, things like cell theory, common components of cells, and the basics of cellular energy processing. Also talked about genetics, the central dogma, evolution, and ecology. These things are universal features of life as we know it. Organismal biology, by comparison, is all about biodiversity, or in other words, how many different ways are there to use these principles to exist as a living organism on Earth. On this slide, you can see just a few of these different ways. Many are not represented, but we're going to talk about all of them. In each chapter, I will open up with a set of learning objectives. Please notice that all of these learning objectives begin with a verb or an action word, and that action should be something you should be able to perform. This is the skeleton of the course. Every question on a quiz or an exam will test your ability to perform these actions, so it's a good starting point for your regular study. I am not going to read these out loud, but at the end of the slide deck, I will show these again a second time. Our first task is to learn some new words and terms. There will be a lot of that this semester, learning new language to describe all of Earth's biodiversity. Speaking of biodiversity, that is our first word, and it means all the different sorts of living things. These things you've heard of before in Biology 1020, such as species, genera, families, orders, classes, phyla, kingdoms, and domains. We might talk about the total biodiversity of an area such as the biodiversity of Lee County, Alabama. Systematics is the study of diversification and relationships between the groups. Lions and tigers and bears are all mammals, which is a class, and they are all in the same order, carnivora. But lions and tigers are in the cat lineage of carnivora, and bears are in the dog lineage of carnivora. In fact, within the cat lineage, lions and tigers are in the same genus, Panthera, along with other so-called big cats like panthers, dodoi, leopards, and jaguars. Trying to resolve these relationships between populations and species is what systematics is all about. Taxonomy is the study of naming groups of organisms and goes hand in hand with systematics. We still don't know about every species on Earth, and organisms are discovered all the time. When discovered, a species needs to be described formally, meaning a description of that organism and how it is different from closely related species, where it has been found, and things like that, as well as giving it a new name. These all have to be documented. This is true not just for species, but all ranks require a published description. This is known as taxonomy, or the science of naming organisms. Any named group of organisms, for example, the species Panthera tigris, or the family Felidae, can be called a taxon. A taxon is any named group of organisms. And because this word has Greek roots, the plural form is not what you might expect. But one taxon plus another taxon equals 2 taxa. Classification is the process of assembling related groups of species into genera, genera into families, and so on and so on. All of these treatments of biodiversity first came about in an organized fashion in the 18th century, the 1700s. And these sciences were very influential on a British scientist of the 19th century named Charles Darwin. I'd guess that you've heard that name before. In fact, the study of classification and systematics and the difficulty in fixing permanent names to species were highly influential on Darwin in developing the idea of evolution by means of natural selection. And in a case of the student becoming the master or the tail wagging the dog, the modern view of systematics is to try and assign organisms to taxa based on their shared common ancestry. In other words, their evolutionary history, which I will henceforth call phylogeny. The father of systematics, taxonomy, and binomial nomenclature was Carolus Linnaeus, also known as Carl von Linné. He was born in Sweden in the 1700s. Linnaeus' world was one where new types of creatures were appearing in Europe from the far corners of the world. And he realized that there was no system to catalog and uniformly describe the known biodiversity of his native region, much less all of the new kinds flooding in with the sailing ships. So he devised a naming and classifying system that we still use today. One aspect of this naming system was that every kind of living thing, which he called a species, should have a two-part name or a binomen. The first part of the name was the name of the group of species that were similar, or members of the same genus. The second part of the name was the specific epithet. For example, Homo sapiens, our own species. Homo is the genus that includes other human-like animals, now all extinct, such as Homo habilis or Homo neanderthalensis. Another example would be Panthera tigris, the tiger. As you can see, the proper way to write a species name is in two parts, with the first word, the generic name, capitalized, and the second word, the specific epithet, in all lowercase. Both of these words should be in italics when typewritten, or underlined if handwritten. The other bequeathal of Linnaeus that we need to learn about is this system of classification. All living things can be put into groups, within groups, also called a nested hierarchy. Similar to how you live in a town, in a county, in a state, in a country, on a continent, on a planet. This town is not that town, though they share a state. And this state is not that state, though they share a country, etc. The same is true for this classification scheme. You can see on this table that tigers and eagles and oak trees are all different species, but at some level all living things are related. All three of these living things are eukaryotes, so they share eukaryote characteristics. Tigers and eagles are both animals. They're both vertebrate chordates, so they share the animal characteristics and the vertebrate characteristics. What Darwin thought was a novel and different idea was that these hierarchical classifications reflected a historical divergence from common ancestors. How do I know what Darwin thought? He told us so. He thought of the nested hierarchy like a tree where each taxon was a branch separating into smaller and smaller branches with species at the tips of these branches. So we can take the information from this table and map characteristics onto Darwin's tree thought diagram. On this tree, you can see eagles and tigers are closer together than either are to the live oak. But live oaks, tigers, and eagles are all more similar to each other than any of them are to things like bacteria. Let's introduce a couple of terms that we will see a lot this semester. Clade comes from the Greek word for branch. A clade is a lineage of organisms that is derived from a single common ancestor and contains every descendant from that common ancestor. Mammals are a clade. All mammals share a single ancestral population of organisms, which we will discuss toward the end of the semester. Every mammal is related by descent from that common ancestor. There are no mammals that are not descended from that ancestor, and there are no organisms descended from that common ancestor that are not mammals. By way of contrast, a grade is a group of organisms that share some similarity in structure, function, or habitat, but that similarity is not because of shared common ancestry. Or it may be the case that the common ancestor has a characteristic, but not all descendants retain that characteristic. An example of a grade would be marine vertebrates. The oceans have plenty of life possessing backbones today. This includes all fishes and sharks, but also whales, dolphins, and porpoises. Whales, dolphins, and porpoises are all mammals, so they are in the aforementioned clade that they share with us humans and lots of other terrestrial vertebrates. Perhaps it is more clear if we go back to Darwin's tree picture. I can draw ovals around branches, or clades, and hopefully you see how it is a nested hierarchy, as Linnaeus forecast. Each oval includes a single ancestor or line leading to the divergence point, and every single branch diverging from that ancestor being inside the oval. Another way of saying the same thing is, instead of ovals, let's take a pair of scissors. We can define a clade, the result of making a single cut, and including every twig that falls out with that branch. This is a powerful way of looking at the diversity of life on Earth. We can apply it at every scale. For example, the broadest level of categorizing cellular life, our three-domain system, looks like this: bacteria, archaea, and our own lineage, the eukaryotes. We'll delve a little more deeply into this very soon, so I won't say too much about it at this point. We can take one of these clades, the eukaryotes, and see finer details of the tree of life. Within the eukaryotes are everything we normally think about as living things: animals, plants, and fungi, or macroscopic life. This representation of the eukaryotes shows that fungi and animals are in a clade that excludes plants, and that plants are in a clade that includes red and green algae, which are also photosynthetic. What does this tell us? Fungi and animals share a more recent common ancestor than either of us do with the plants. Again, this is something that we'll delve into over the course of the semester. We can zoom in even further, this time within the kingdom of animals, within the subphylum of vertebrates, within the class of mammals. Now I'm saying that this same tree represents the mammalian order Carnivora. Within this order, we find many families, genera, and species, including lions, and tigers, and bears. Did you say it? Oh my. But these three named groups of animals, or taxa, are not the only members of the order Carnivora. Carnivora includes many other animals, including house cats, seals, sea lions and walruses, dogs, foxes, skunks, and many more. Why are the lions and tigers shown on this branch and bears and dogs on this other branch? Because of the shared common ancestry of all carnivores diverged into two groups, which we recognize as the cat-like lineage, Feliformia, and the dog-like lineage, Caeniformia. Now that we've seen some of the basics of how tree thinking can be applied to biodiversity, we need to learn some terms to describe clades and things that look like clades, but maybe they aren't. A true clade, which includes a single common ancestor and every single descendant from that ancestor, is monophyletic. Mono means one, like a monocle, and phyletic refers to the evolutionary history of a lineage. Holo means all, or the whole of something. So a holophyletic lineage is essentially synonymous with a monophyletic lineage. Para means on the side of, or adjacent to. A paraphyletic lineage has a single common ancestor, but doesn't include all descendants from that common ancestor. Paraphyletic groups are thus not clades. Poly means many, and a polyphyletic trait or lineage refers to a group of organisms that may share some characteristic, but that characteristic evolved multiple times independently. And therefore, polyphyletic groups are not clades either. All these terms are probably just abstract word salad, so let's try to apply them to some concrete examples. Here is a more modern version of Darwin's tree diagram, also called a phylogram, which shows the evolutionary relationships between the animals with backbones called vertebrates. The vertebrates are both monophyletic and holophyletic because every vertebrate animal is represented in this diagram, and no vertebrates are found in other parts of the animal kingdom. In other words, there is no butterfly or jellyfish that also has a backbone. Our mammal lineage diverged from the reptilian lineage before the ages of dinosaurs began in the Paleozoic era. We share a monophyletic grouping with the reptiles called the Amniota because we share anatomical features that allow us to live a life independent of laying eggs in bodies of water. You may notice something unusual about this representation of the reptiles, which are represented by this yellowish shape. If I were to ask you to name some familiar reptiles, you might say things like tortoises, cobra, gecko, iguana, alligator. But if I said penguin, you'd probably raise your eyebrow and back away from me slowly. But see here, birds are part of the reptile clade, only they've diverged so much from the rest of the group of reptiles that they are unrecognizable as reptiles. Thus, the traditional group of reptiles, which includes a single common ancestor and many, but not all descendants of that common ancestor, is paraphyletic. If this shape were an actual tree and we wanted to prune out the traditional reptiles, we would have to make 2 cuts. If we wanted to prune out all amniotes or all tetrapods, we would only have to make 1 cut. Now let's talk about this red shape here, the polyphyletic shape. Polyphyly means something has evolved multiple times. So what characteristic do we see that evolved in both mammals and birds? There are a couple, but let's focus on 1 or 2. Both birds and mammals regulate their own body temperature independently from their surroundings, or we are warm-blooded, though that term isn't the best descriptor. Another characteristic found in both birds and mammals is a 4-chambered heart. Fish have 2-chambered hearts. Amphibians, like frogs and salamanders, have 3-chambered hearts, and so do turtles, lizards, and snakes. Then, crocodilians have a 4-chambered heart, but with a modification that we don't see in birds or mammals. The simplest explanation for the presence of 4-chambered hearts in both birds and mammals is that the character evolved twice independently. So the common ancestor of birds and mammals, the earliest amniotes, did not have a 4-chambered heart. Okay? Here are a few more terms that we need to introduce to talk about how diversity has evolved. Characteristics that are ancestral or in the older of 2 lineages are called plesiomorphic. Apomorphic characters are newer or more recently derived. If an ancestral character is shared by different organisms, we call it symplesiomorphic. If a derived character is shared, that's a synapomorphy. We will talk a lot about synapomorphies over the course of the semester, as synapomorphies are the best tool to help us identify monophyletic clades. One more term we can use is autapomorphic. This describes a character that is unshared but derived. More concrete examples follow in a moment. A couple of terms that should be review for you in your discussion of evolution are homology and analogy. Homologous structures are similar based on shared common ancestry. For example, the forelimbs of whales, bats, tigers, eagles, and you and I, only we call them arms, share the same pattern of 1 bone, 2 bones, many bones as they extend out from the shoulder. Even though a bat's wing and a bird's wing are not the same as a tiger's claw or a whale's fin or your arm functionally, the underlying structure is very similar, and that's because our common ancestor had it that way. Analogy is similarity based on common function, but not due to a common ancestor. A bird and a bee both have wings, but a bird's wing is a modified forelimb, and a bee's wing is an entirely novel structure with no bones at all. The shared common ancestor of birds and bees, which was an invertebrate with bilateral symmetry, did not have wings. So this is a case of convergent evolution. Another word to describe the wings of birds and insects would be to say that they are homoplasious. Let's return to this phylogram once again, this time to apply the new terms. This phylogram shows the evolutionary relationships between the vertebrates. First, let's outline some ancestral characteristics. All vertebrates are animals, which means being an animal is an ancestral characteristic or plesiomorphy. It is shared by every vertebrate, and sharing means it is symplesiomorphic. We could list off many other kinds of symplesiomorphies. Vertebrates are all multicellular. They all have DNA and ribosomes and mitochondria. They all breathe oxygen as a way to produce great volumes of ATP. They all eat with mouths. These are all symplesiomorphies, and they don't help us to establish relationships between the animals within the phylogram. To know where to put these animals in relation to each other, we need to identify derived characteristics, apomorphies that tell us where novel features became the bases for branching. What made amphibians and all other vertebrates distinguishable from fish? Legs, 4 of them, which gave us the clade tetrapoda. Thus, relative to fish, being a tetrapod is a derived or new characteristic shared by amphibians, reptiles, birds, and mammals. Being 4-legged is a synapomorphy. What makes mammals, reptiles, and birds different from amphibians like frogs and newts? We share many derived characteristics, including the presence of an egg that doesn't require laying in water, the amniote egg. This is another shared derived characteristic, or synapomorphy. Another way of saying a feature is new, relatively speaking, is apomorphic. But new in the case of tetrapods is about 400 million years ago, and new in the case of the amniote egg is around 320 million years ago. So new is not the word we want here. Instead, let's say apomorphic. How about an autapomorphy? An example of a derived character that is unshared, only found in a single lineage. An example of an autapomorphy in this phylogram would be feathers. Every species of bird, and there are about 10,000 species worldwide, if Wikipedia is to be believed, has feathers. But how do we determine the closest recent ancestor of birds in this phylogram? No other lineage has feathers. So feathers are not a useful characteristic for finding the relationship between birds and other tetrapods. How do we know where birds fit into this phylogram? We need to look for other characters that may be shared but are also derived. Hair is kind of like feathers in that it's made mostly of the same protein, keratin. We also share 4-chambered hearts and homeothermy, or warm-bloodedness. So why aren't we mammals drawn as sharing a clade with birds? Because a mountain of other evidence shows that these characters are not homologous or similar by shared common ancestry. These characters are analogous or similar, but not because the shared common ancestor possessed them. Analogy and polyphily are not exactly the same thing, but they often go together. And when characters are this close in appearance but analogous, we call them homoplasious. You should remember the term convergent evolution as the appearance of analogous characters that often give a false impression of membership in a clade. The branching that Darwin hypothesized and that many subsequent lines of evidence support is divergent evolution. The divergent characters are apomorphies. Earlier on, we learned about these 8 levels of taxonomic grouping. Every species can be placed into this context of belonging to every one of these levels. Even if there is only one still living or extant species in a whole phylum, which we will encounter this semester, it still belongs to a lonely class, order, family, and genus. But sometimes we have the opposite problem. Some taxa are over-rich in diversity, and in that case, 8 is not enough. We can generate groups of these groups, or subsets of them, by adding prefixes or like super, sub, or infra. Within a very large family, we may define a group of genera that we call a tribe. Let's look at an example. What is the most successful group? By most metrics, it's the beetles. But not just these beetles, but also these beetles, which I was at an embarrassingly advanced age before I realized these words are not spelled the same way, I must admit. The beetles represent a monophyletic order, Coleoptera, with over 1 million described species. This is within the most diverse class, the insects within the most diverse animal phylum, the arthropods. It will take us most of the semester to get back to talking about the arthropods, but let's look at one species of this very successful order. The southern pine beetle is one species that is found around the southeastern United States, from Virginia to Texas. You've probably never seen one of them. As you can see from this illustration, they are very small. They also live almost entirely inside the bark of southern pine trees. You may have seen a patch of dead pine trees that were killed by swarms of these beetles. So why do I mention them now? Because of the information on this slide, taken from bugguide.net, an online entomology resource. The southern pine beetle is a member of the largest family of the largest order of the largest class of the largest phylum. And as such, you can see that we need every available taxon, prefix, and even an extra one, the series Cucujiformia. We won't get into every one of these groups, certainly. This is just one example to demonstrate the awesome task ahead of us, describing the great diversity of life on Earth in about 4 months. We will begin at the very top of this hierarchy, which is mercifully less complicated than Dendroctonus frontalis. There are only 3 domains, and we will dispense with 2 of them rather quickly, the bacteria and the archaea. Once again, here are our learning objectives, and in our next discussion, the prokaryotic lineages.